Good afternoon, everybody. I'm glad to be here.
Take your time.
Yeah. I want to thank Dr. Gemma for the invite. And today I will talk you about our work on HIPK2, that is the homodomain interacting protein kinase 2. So I'll give you a brief introduction in the gastrointestinal pancreatic organoids with a brief history. Then I will talk you about our data on our on the HIPK2 role in the pancreatic cancer. And then I'll give you a short introduction in a new three-dimensional culture that we are employing in our experimental design. That is the organotypic tissue slices. So, what is an organoid? Here are some definitions. If you ask ChatGPT, it answers that an organoid is a miniature, simplified version of an organ that is grown in the lab from stem cells. That is a vague but quite good definition. And what you have to keep in mind is that an organoid is a stem cell culture. So differently from tumoroid or spheroids, the organoids originated from a stem cell. And indeed, what Hans Clevers and his collaborator originally discovered, Hans Clever is the pioneer of organoids culture, is that a particular protein, that is a membrane receptor, LGR5, is actually an epithelial stem cell marker. Indeed, you can see in this experiment they performed that you have LGR5 tagged with the EGFP, and in the intestine, LGR5 expressing cells localized at the crypti of the intestine, and with a gray reporter. So here you have the cells that originally expressed LGR5. You can see as all the cells of the villi are originated from the LGR5 expressed cells. So that LGR5 demonstrate that LGR5 is a marker of the staminality of these cells. And mechanistically, what happened is that our spondin binds LGR5 receptor, and this is crucial for the degradation of RNF43, that is a ubiquitin ligase that can degrade and eliminate the Wnt receptor and the Wnt signaling. So actually what happened when our spondin binds LGR5 is that the Wnt signaling is persistently active, and this is crucial for the epithelial stem cell, for their maintenance and their proliferation. And based on this, they were able to define a media containing Wnt and spondin or other growth factors to establish organoid culture from the intestine. Then they expanded their findings to other tissue like colon, stomach, pancreas, and liver, and so on. And in particular for the pancreatic tissue, here you have a normal histology of the pancreas, in which you have the acinar parenchyma, the duct, and the Langerhans islet, that are the main components of the tissue, pancreatic tissue. And they demonstrated that in collaboration with Tuveson Lab, in this tissue, the LGR5 expressing cells reside in the ductal cells. Indeed, the first thing you have to do to establish a pancreatic organoid culture is to isolate the ducts. And this is in the original paper, they say that they handpicked the ducts, and it was very hard to perform. Now there are protocols that much easier way you can get your ducts. You can put them in matrigel, and you can cultivate them with Arsponian wind media. Of course, you can start from murine or human tissue. And what it was interesting that in collaboration with Tuason Lab, they established organoids from a classic model of pancreatic carcinogenesis that is based, a murine model, that is based on the KRAS G12D expression. In pancreatic cancer, the 95% of the patients carry KRAS mutations, and also in mouse, if you express KRAS, and here you have the pancreatic specific expression that is obtained using this transgene, that basically you have a stop codon flanked by LOXP sites that is upstream the KRAS G12D allele. And then you express the CRE recombinase under the control of PDX1 promoter, that is a pancreatic specific promoter. And so you obtain the expression of KRAS G12D only in pancreatic cells. And after a few weeks, you obtain, this is the normal tissue. After a few weeks, you obtain metaplasia of the tissue with the neoductologenesis, so the new ducts are formed. And then you, after a few months, you obtain the pancreatic neoplasia of the ducts. And if you combine also the p53 mutation to this system, you can easily obtain pancreatic adenocarcinoma samples and metastases. So what they did is to establish organoids from this lesion, so from panine, from tumor, from metastasis. They demonstrated that the karyotype of these organoids is similar to the tissue of origin, is stable in time. And if you transplant these organoids in syngenic mice, what you get is a similar lesion to the tissue of origin. So they maintain also the pathologic feature of the tissue of origin, the lesion of origin. And so to conclude this part, what to me is the pro of the organoids culture is that they are easy to establish and they're easy to be manipulated. They are genomically stable, they harbor differentiation potential, and they resemble physiologic and pathogenic features. And moreover, they are a great way to reduce the experimental animal use that in Italy is a problem because we have a lot of restriction to perform experimental animal to use animal in our experimental design. And the cons, of course, is that the organoids are costly because you have a defined media and the matrigel, of course. To me, the problem is also compared to a cell line, we have a low amount of biological material. So, for example, to perform a Western blot is not so easy. And of course, they are not as heterogeneous as the tissue samples. What we have done with the pancreatic organoids. In our lab, we study this kinase that is the homodimine interacting protein kinase 2. That is a kinase widely implicated in a lot of cellular processes and molecular pathways. In general, we can say that is a kinase that is activated in response to stress, and is well known its involvement in the DNA damage response. When I started my investigation on HIPK2, we were interested in the HIPK2 role in cell division. Indeed, what we observe is that in HIPK2 knockout, murine fibroblasts or in HeLa interfere for HIPK2, the cytokinesis fail, and you obtain tetraglobalization, so vinucleated cells and tetraglobalization. And what we also observe is that in transformed MEF, murine embryofibroblasts, transformed with E1A and RAS oncogenes, this tetraglobalization event associates to an higher tumorigenicity and an higher chromosomal stability. So, starting from this, we wondered if the HIPK2 loss or downregulation might be an early event in the pancreatic carcinogenesis, considering that pancreatic cancer is among the tumors that are more characterized by a near tetraploid karyotype cells. So we adopted the KRAS G12D mice model, and we crossed the mice with an HIPK2 condition knockout mice. So we got mice that in pancreatic cells were knockout for HIPK2, and simultaneously they expressed the KRAS G12D in a pancreatic specific way. And so we established the organoids, and we checked for tetraglobalization of enucleation. But as you can see, in our KRAS G12D HIPK2 knockout organoids, the percentage of enucleated cells was very similar to the KRAS G12D control. So, and also the nuclear area of the pancreatic cells from the pancreas biopsies of our mice was similar to the control. So nuclear area is an indirect way to measure DNA content and so tetraglobalization. So and moreover, so for some reason in pancreatic tissue, the cell division is not regulated by HIPK2, and moreover, what we observe in terms of carcinogenesis is that in the KRAS G12D HIPK2 knockout samples, we have a slowdown of the carcinogenesis. Indeed, you can see here in the KRAS G12D sample, you have a lot of duct, and you have also ductal with neoplasia, whereas in the KRAS G12D, in the knockout, you have less ducts and with a pathologic rate that is lower. So this is an opposite result from what we expected, because we expected an acceleration of the carcinogenesis. And so we have to rethought the HIPK2 function, because in this case, HIPK2 was not acting as an oncosuppressor, but in an oncogenic way. So at that time, several articles were being published on the HIPK2 crosstalk with KRAS, And indeed, what also in our lab was demonstrated is that HIPK2 can cooperate with KRAS and can sustain the KRAS signaling in colorectal cancer progression. And so we checked if also in pancreas this would be true. And both Western blot analysis and immunohistochemical analysis show that there's a reduction in the phosphorylation of ERK, and so in the KRAS signaling. And so the next step was to use an APK inhibitors, that is 5-iodotubbercilin. So we tested if also with a kinase inhibitor we can obtain similar results. And we treated the KRAS G12D mice 2 weeks before the age of the expected panine appearance and for the following 5 weeks. And we obtained very similar results to the genetic model. So the percentage of the neoplasia was reduced, and also the phosphorylation of ERK, and so the KRAS signaling was reduced in the inhibited tissue. And a referee raised a concern about the specificity of the 5-iodotubbercilin. Indeed, the 5-iodotubbercilin is quite a specific kinase inhibitor, so he asked us to perform the treatment also in the knockout KRAS G12D mice, but for the restriction I said about animal experimentation, we cannot did the experiment on mice, but we decided to test it in the organoids. And so we treated with the 5-iodotubbercilin both KRAS G12D and KRAS G12D HIPK2 knockout organoids. We induced the KRAS signaling with murine AGF, and then we checked for phosphorylation, and you can see how the phosphorylation of ERK is increased only in the KRAS samples and not in the KRAS HIPK2 knockout samples. And this is another story. So to make a long story short, another important function of KRAS, oncogenic KRAS in the pancreas is that is able to reshape the microenvironment of the pancreatic tissue. And what was interesting in our sample is that we observed a different kind of microenvironment in an HIPK2 regulated way. So we actually have seen that in HIPK2 knockout associated with a peculiar microenvironment that is characterized by an higher collagen deposition and a lower positivity to alpha SMA compared to the KRAS G12D. And so what we have concluded with our work is that the loss of the inhibition of HIPK2 can reduce the KRAS signaling in KRAS G12D pancreatic sample, and that also we also observe and characterize this particular microenvironment. And then I will jump to another project. Then since we have studied fibrosis and collagen deposition, and we have developed protocols and tools to investigate on this. What we have decided to do, since fibrosis is a common feature of many cancers, and what has been observed is that in many tumors you have an increased stiffness due to the fibrosis process, and since HIPK2 is also a regulator of fibrosis, indeed is a master regulator of kidney fibrosis, and is important also for cardiac fibrosis. We decided to test, we start another project focusing on hepatic fibrosis. Hepatic fibrosis is a big issue for health system because liver pathologies are very common in worldwide, and so there's no cure or effective therapy for prevention for fibrosis. Basically, the pathology of the liver starts with an injury that can have different sources of alcohol abuse, viral infection, excessive drugs, metabolic dysfunction, can injure the liver cells. And so there are a multi-step process in which you have inflammation, you have the activation of collagen producing cells, and you have if this deposition of collagen is excessive, you have the fibrosis, and fibrosis is actually a physiologic process. So is what the organ do to fill the hole that the damaged and dead cells live in the tissue. So it's something that the organ, it's a response to maintain the structure of the organ, and it's a reversible process. So if you eliminate the cause of the injury, the tissue can regenerate in their original, in its original form. But the problem is that if you have a chronic injury, you can develop cirrhosis, that is the irreversible form of fibrosis, and cirrhosis is highly associated to hepatocellular carcinoma. So we decided to, what we are doing in our lab in this project is investigating on the HIPK2 inhibition as a therapeutic approach for fibrosis treatment, and we are trying to identify new biomarkers and new possible therapeutic approaches. And so we are developing in vitro model, collecting organoids, in vivo model, developing fibrotic murine model, and an ex vivo model with this new organotypic culture. And for the in vivo model, there is the Silvia Sozzi's poster presentation. Briefly, what we have done is we have crossed the HIPK2 conditional knockout, the same from the pancreatic model, to an albumin expressing mice. So in this model, of course, the expression is under the control of a liver-specific promoter, that is an albumin promoter. So we obtain the HIPK2 liver knockout mice, and we induce the fibrosis in two different ways: by carbon tetrachloride injections, that is a classical way with chemicals that is able to damage the liver cells, and with the choline deficient diet, that is a particular diet that drives the fat accumulation, and so is able to mimic the statotic, the fat accumulation, the statotic process, and the fibrosis accumulation. And now we are collecting the samples, and we are performing the analysis. And what we are also doing in parallel is to generate tissue slice cultures. And basically, you have a slice of tissue in plate, and this is quite easy to obtain with this instrument that is called vibratome. And so you, starting from a liver biopsy, you embed the tissue in agarose, and with the vibratome, you can cut the tissue in 250 micrometer thin slices, and the vibration of the blade happens at a frequency that does not damage the cytoplasm. So you have living cells in your slices, and so you can take the slices, put them in a dish with the millicell inserts, and cultivate them in media. You can treat them for a few days and weeks. And this is actually the work of Clizia Maccaroni, that is the postdoc of our lab. And so this ex vivo model of organotypic slices is a good model because it preserves the architecture of the cultured organoids. So you have all the components of the tissue, including stromal cells, including immune cells. And so there are an excellent biological surrogate for different pathologies and for short and mid-term analysis and treatments. And what we are doing with these slices, we have two different approaches. One we get the slices from the murine samples, so the fibrotic samples. So after the CDA treatment, we collect the biopsy, we get the slices, and as you can see, here you have the collagen staining in liver tissue biopsy and the normal control, and here the same staining in the fibrotic slices after 3 days in culture. So you have a similar, you can maintain the structure and the collagen presence in the sample. And we are now treating these slices with the different kind of HIPK2 inhibitors to see the response in terms of collagen expression and other pro-fibrotic markers. And the other approach is on human sample. So we get normal liver biopsy from the surgery. We get the slices from this biopsy, and we induce in these samples the fibrosis by TGF-beta treatment. That is TGF-beta is a potent inducer, pro-fibrotic inducer, and so we will see what happened after the induction of this process, analyzing by single cell RNA-seq these samples, and that's all. So I want to thank all of you for the attention, and the head of the lab, that is Silvia Sordu. Cinzia Rinaldo, that is the PI of the pancreatic cancer project, and in particular Crizia Maccaroni, that is the postdoc that is working on the tissue slices, and Silvia Sozzi, that is the PhD student that is working on the in vivo model and all the molecular analysis that we are performing.
Thank you, Davide, for this great presentation. Questions?
I'm playing too, but I can see. Davide, thank you very much for this. I have a question for you, but also for the audience. Let's put us in this situation. You have your mice. You already organized and performed all the genetic modifications, etc. So from the starting point, when you extract the colon,
the liver.
Yes, the liver, yes, sorry, the liver, until the organoid, it took or it takes how much time?
For the liver, the protocol is basically the same of the pancreas, because they have both a similar origin, and so yeah. So this is the pancreas, but it's the same. You have a 5 hours protocols to isolate the ducts and to put them in Madrigal to cultivate them. And after a few days, actually, maybe a week, you have a good confluency of organoids, so you can start with your experiments, you can freeze them. And the important thing to keep in mind is that the organoids can be expanded in a limited way. So you can, differently from my primary cells, you can arrive to passage 50, and they maintain the same.
So you cut them or
you distillate them with collagenase, dispase, and
we put them again in a way to
no, the protocol, as I said, the initial protocol was to unpick the ducts. So you, starting from the digested material, you went under the microscope, and with the pipette, you unpick the ducts. Now you have just to digest the material for 20 minutes, and then recover the supernatant that contains actually the ducts. So you have different time points.
Easier than primary culture.
Very easy. It's very easy to establish.
Okay.
Yeah, and they are also expanded whenever you want passages.
Focus more on organic and produce
I have a question.
Oh, yes.
I'm also confused. No, more seriously. I love that you use multiple models, right? So I think it really it sort of brings home the message of what you're trying to say, which is that it's a fit for purpose. In one size doesn't fit. What I wanted to know from you is, what is it that you find that organoids brings to your research? How does it fit in? How does it how does it help you evolve research? You know, you were using two-dimensional models of primary cells and mice, and you now added organoids. So what does organoids bring into the lab that was missing?
Of course, they are a great source. To me, for the pancreatic project, was a great source. So because a normal or near normal cell line or primary cell line, primary culture from pancreas don't exist. So there are just maybe few cell lines, and the primary culture is very hard to obtain, and the organism very easy to obtain. So to me, it was a fantastic revolution. And from an experimental point of view, you know, you can, as I show you, you can skip the animal part. So you can reduce the use of animal, and you can perform maybe a pilot study or a pilot experiment in a very easy way, in a very fast way. So this is for me the great advantage.
So what is the best model tools?
Okay.
So what is the best tools used and the advantage when using these tools? Vivo, ex vivo, or in vitro?
Which is the better?
What is the best?
I don't think there's a better model or better tools. The point is that you have to answer the same question with different approaches, and you have to find the right model to answer your biological question. So I don't think there's a better model or a better tool.
Okay, so thank you. My question is, in fact, I'm working on muscle fibrosis in muscle dystrophy. And my question is always when we use a certain compound, we can sometimes reduce fibrosis, but it doesn't shift the monocytes, the macrophages, did you check on your model if the macrophages did change the behavior, immune behavior?
For the pancreatic project, we checked by F4 80 plus antibody, but we didn't see any difference in the fibrosis, hepatic fibrotic model. We have to perform the analysis, but we have also the single cell RNA-seq, so we will get some insight from this kind of analysis.
Thank you for the presentation. My question is, can we use the pancreatic organoids to study the genetic mutation associated with type 1 diabetes on beta cell function and obviously the immune system interaction?
For diabetes, you have to consider that, as I showed you, the pancreatic organoids are actually an epithelial cell culture. So for diabetes, you need the other components that are the Langerhans islet, that are of course the cells in which you have all the endocrine production. So for diabetes, there's some, I think, some primary culture from the islet, but I haven't tried to obtain. We were interested in the ducts, and because the pancreatic cancer actually start from the ducts, from the ductal cells.
Congratulations for the outstanding results. But my question is a technical one, then Mohammed's question. As I worked on gastrointestinal, I'm interested to know the minimum requirements for launching such experiments on 3D models. Because as you know, we are in mixed countries, so we need the minimum requirements to launch such experiments. Such as the bioinformatics, infrastructure, and human resources, etc. Thank you.
Minimum requirements, you mean equipment?
Yeah, the minimum one.
But actually, you just to have a biosafety cabinet, bath, because the digestion is performed at 57 degrees, and what else? The enzyme for the digestion, so collagenase, lipase, and whatever.
I would say maybe
Just to do it.
And the night market.
Can I plant my business for a second?
Yes.
So on the Merck website, on the Merck website, we have our 3D Soul Culture Learning Center. In the Learning Center, you will find a whole set of technical articles just about how to establish basic organoid culture and our recommendations for what that looks like in terms of reagents, and also we're in the process of doing a how-to video, which will be put up probably in January. But if you want to know, there's a full list of reagents for basic organoid culture, including dissociation, cryopreservation, dome, plates, the whole thing is there on our website.
Yes.
I would like to add, what are the challenges you have faced in targeting the HPPC for
What are they?
challenges?
The challenges.
Yeah, in targeting the
HPV.
it's HPPC, yes, for the research on
The challenges is that I skipped this part, but HIPK2 is a complex protein for many reasons. First of all, is two, because there are other IPK. So there are IPK1, IPK3, IPK4, and they have some homology. So the big challenge is to make sure that you are inhibiting or you are observing just the HIPK2 role. So we are using inhibitors for all the HIPK. So the knockout is only for the HIPK2, but the inhibitors are for all the HIPKs. So we have a problem resolved, and the challenges are also that it's a protein involved in a lot of molecular processes. So you focus on one process, but you have to keep in mind that there are DNA damage response, nutritional stress response, and the other kind of pathway that could be cell division that could be affected by the inhibition.
I use my position as also co-organizer to forge in.
You can let me give you the time to say that we are over now.
We are
Because
over. Last question.
We have to go
Last question.
break.
For you, Davide, and for also to the audience. So we talked about the cryo the conservation of organisms.
Yeah.
Is it the same protocol if we? Yeah. So it is the same protocol cryopreservation organoid and cell line.
I can say it's the same, but you have to consider that they are stamina cells. So they are, compared to tumor cell line, of course, they are more sensitive to the freeze and thaw.
Okay.
So you have to, we use freezing medium,
Yeah.
but it's basically a medium.
We have
No, we don't. I agree. The other thing I'm going to say is things on the organoid. So we've done a lot of work around cryopreservation on organoids. We have a shipped biobank, right? So one of the takes is cryopreservation and then resurrection from cryopreservation. And what we found is that you have to optimize to type. So for example, colonic organoids resus better if we fragment and freeze, whereas PDACs are better if we freeze whole. So that's the one caveat I would say is, and we recommend the DMSO-free,
Yeah, this is true.
free.
Actually, what we have seen is that if you digest the organoid before the freezing, is better. So you have an higher viability.
Well, for organoid culture,
You have to taste it.
you.